This wants me to test my microphone. Oh. Okay, well, they wanted me to test it. Good evening. Thank you for joining me tonight. Um, I don't know why my computer was just like, let's test your mic and your speakers. And I was like, okay. So welcome on in. So um I'm just gonna say, because I already have your guys' student ID number and you guys don't need to keep sending it to me, I'm just going to look at who's here and then rewrite your um your CRN, or not CRN, your ID number, and you guys don't need to retype it because I have your names and ID numbers next to it and you're good. But if you are new here, none of you guys are, but if anybody comes on who's new, I'll make sure to give them the rundown and ask for that. So let me write your guys' stuff down before we get started. And it's going to always probably be a 1 and 2 part Zoom meeting for anything that's over 30 minutes, because Zoom wants to make me pay for anything longer than 40 minutes. So what we'll probably wind up doing is when we take a break, I'll have you guys in the meeting and then come back after the break and rejoin the meeting. So it's technically a whole new meeting, if that makes sense. Let's see. Did you guys happen to get my text for the review material? That is some stuff from Caesar. He was a pal for me. So that was some stuff that he sent out during last semester. There is a disclaimer on all of them, or at least there should be, that none of those questions are going to be on any of, they're never going to be on an exam. So any of those practice questions never going to be on an exam. So I just wanted to clarify that. They're more so for you to ask yourself, do I understand this concept? So if you find yourself having to look up, thank you, Melissa, by the way. But I will, if you've come before, I'll be able to just rewrite your name and your CR and your at number. But anyways, for the sample questions, it's more so if you are sitting there and you're stumped on the question, it's a whole topic that you'll need to review, not just memorizing what the answer is to that specific question, if that makes sense. So that'll be how you use those study tools. So I know, Amanda, I believe you're on Tuesday, Thursday, but you came in on Monday too.
I'm in Monday, Wednesday now.
Okay.
I've moved sections, so yeah.
Because I remember seeing you Tuesday, Thursday, and then I also saw you Monday, and I was like, what is happening? Like I can't put my finger on when I'm going to see this. But was it just like a scheduling conflict that was
Yeah, because I also am currently taking Bio 33, and so I felt as though it'd be better. And I also do SGA, so like every other Wednesday I have meetings, so I was like, I have to figure this out. I have to, get out of class by 4:30 every other Wednesday. So yeah.
Okay, well, then yeah, that would make sense. So I'm glad that worked out. But so who else is in Monday, Wednesday? Because that means that you guys had the most recent lab today. I know Ling Ling, you're in Tuesday, Thursday. If I'm not mistaken. Yes, Tuesday, Thursday. But I'm sorry. Is it because I know sometimes it's different. Is it Elisa or Eliza?
It's Eliza.
It's Eliza. Okay. And then Jamie, I can get, and Melissa. So Tuesday, Thursday. So if we review anything, because I understand that, you know, this is scheduled where it's Monday, Wednesday, and then Thursday before lab. So Tuesday, Thursday, people, you might be sitting here like, what's happening? But I promise we'll go over like old stuff, and we'll also kind of preface new stuff. I'll try to do that. But is there, so I know Monday, Wednesday, you had your new lab, and you guys should have done the gram stain, if I'm correct. Okay. So, okay, good. Okay, I'm glad that it's helpful. Because I understand that, you know, you guys might sit here and you're like, what is happening? But I'm glad that it prefaces the lab a little bit better. So the gram stain, are we okay on that? Okay, yeah. And I know, yeah, I can explain the steps. Okay, so let me go ahead and open up because I want to make sure that, you know, I'm wording it the same, because you know how exams are. She's going to use the same wording she does in the lectures, or very similar. If she wants you to focus on certain words, you know, kind of like with the biosafety levels, some of them had the potential to be hazardous and some of them were hazardous, you know. So let me open up some stuff. So I'll go over the steps of the gram stain. And was there anything else, Monday, Wednesday peeps, that in the lab that we thought that we wanted to go over? I'll double check the chat in just one second after.
I was just gonna say, I feel like something that was like important was just like remembering like the shapes of like the bacteria that you're looking at. So if we can go over that again today, but I know that we go over that a lot, I feel like in all the PAL sessions. So
yeah, we could go over it again. There's no shame in it. Even when we review the gram stain, we'll look at it a little bit, because I'm sure that there's going to be a few things on here. Let's see. I have cell culture, results, staining. Here we go. So obviously with a gram stain, you're going to be able to see arrangement and cell type, right? So off the top of our heads, what type of cell types do we have? Because that's in the same thing I have. Can we sorry, do you guys know any yes, any of the shapes? Sorry. Sometimes they're referred to as type, but yes. How does it, any on the Chrome Dome that you guys know of? I don't mind if you type or unmute yourselves. It's bacillus, good. And what's another name for bacillus?
Rod?
Yes, good. We have the cocci, and then we have one more. There you go. It's going to be called spirochete. I know it's strange, but spiral, you'll get the gist. And what is the one that that's, I think we have over 300%. We have like 300% times the like contraction rate in Bakersfield. What is the spirochete STD? That might have been a little too much, but so not quite fungus. Syphilis, spirochete. There you go. So fungus is going to be a type of eukaryotic organism. And remember that bacteria is more often than not prokaryotic, right? So our three types are going to be cocci, which are the spherical ones, and then we have our rods or bacillus, and then we have our spirochetes. Now, I want you guys to think of the cocci and the bacillus as brother and sister, right? They're very closely related. They may look a lot similar. They may look really similar. And then spirochete's like the weird adopted cousin, right? So spirochete is usually going to be by itself. It's not going to have a whole lot in common, and it's just kind of over there. But with the cocci and the rods, they're going to have similar names for their arrangements, right? We have diplococci, we have diplorods, or diplorods, or I think, let me double check. Diplobacilli, sorry, diplobacilli. So we have our, it's coccus bacillus, diplococci, diplobacilli. We have staphylococci, and that's Staphylococci is the only one, I think. Yeah. But we also have streptobacillus and streptococci, right? So they have similar morphologies in the end. So let me go ahead and share my screen really quick with you guys so we can go over this. Here it is. Okay. So you can see what I mean, right? So we have 1, 1, 2, 2. And then that's kind of where we finish with the twos. But then, what if we get a chain of 3? Chain of 3. That's going to be our strep. Now, cocci goes to 4, which is our staph, right? And tetrad. I guess you can switch those around. But these are going to be our main arrangements, right? Spirochete, if we look at spirochete, it can just be kind of like shorter, or it can be longer. It's not really like, it doesn't have any really special name if it's any longer, or it doesn't have like, if it's like curved a certain way, because remember, they move. They're going to, they might not always look as straight as, a very straight. They could almost look like the spirochete picture here, right? So that's how I kind of want you guys to think about those three. As you get more familiar with, the gram stain and viewing them under the microscope, you'll become more comfortable also, defining cell morphology and arrangements. So, obviously we went over these in class a little bit, but there is one in between one. And it's like I said, these are very similar to each other. So there's one in between one that's like half and half. We can call this like the parent, I guess, but it would be the diplococci, right? Or not the coccibacillus, I think. Yeah, coccibacillus. It's not on this slide, but it's this example in the bottom right. That's our coccibacillus, because it's kind of looks like a rod, it kind of looks in between. but that's kind of like out there, right? Most of the stuff we're dealing with is going to be strep, staph, some kind of bacillus. It's gonna be different. So you guys, if you're given a hard one, it's gonna be in the classroom, not on an exam. It's going to be pretty obvious on an exam. So obviously we have our strep. What I like to think of for our strep versus staph is kind of like, think of like, stripping the bed sheets, right? Staph, strip, kind of. Well, that sounds pretty close. Or no, it's strep. Strep, strip, strep, strip, right? I'm sorry, I'm mixing up these words. I swear to you, I'm not trying to. Strep, strip, strep, strip. The I and E. Remember the I and E. But I think of that like, like, what is the movement you use for like taking out the bed sheets, right? When you're stripping the sheets, you kind of do that long movement, I suppose. That's what I think of. If you guys have a better one, let me know.
I think of like strep, stripe.
Strep,
Right.
stripe. Yeah, that's a good one. That's a good one. That's better than what I have. I know mine was kind of far-fetched. And then staph, I just think of grapes. I mean, grape has an A, so I think of that. Grape A. So it looks like a bunch of grapes. I don't know. If letter association is your thing, or word, or like something that sounds similar, think of these things and you will be able to remember it a lot better. So that's how I kind of remembered the arrangements. As it goes on, obviously, we're not too deep into the semester, but exam 1's around the corner. It's gonna hit you like a ton of bricks if we're not careful. Because remember how the semester's organized, right? Every 4 weeks is gonna be an exam. So it's barely week 4, yes, but we're gonna have an exam. So as we get past probably the first exam, you're going to feel more comfortable with saying, okay, yes, that is staph, that is strep, that's diplococci, that's a tetrad. You'll be able to become more comfortable with those things. And if you want more practice, we have bacteria slides in the classroom, and you can stay afterwards. I'm not sure on how many days, but if you ask Dr. B, she's more than willing to let you, especially if she ends class early. You're more than willing to, or more than welcome to stay. And take some of those bacteria slides. You can, if you really, really want to, you can take the slide, and you don't look at the little label on the side, place it under the microscope, and then kind of try to test yourself and look away from it and say, okay, I think this is streptobacillus, right? And then it'll have the little name card on the side, and it'll tell you what it is. So if you want to try to test yourself like that, it can give you a little more practice. And if you don't understand it, either me or Dr. B might be there to be like, okay, well, this is why you were seeing that. Because you might think it's diplobacilli, but it's really just two bacillus in between their reproduction phase, right? They might just still be connected. And at the time, they were in the middle of replicating, and we just killed them in time for them to finish, before they were able to finish. So we have to look at what the greater population is, right? And that might be difficult, but I know you guys can do it. I promise with more practice, it'll come easier. I know some people are able to look through the microscope and look at the board and say, this is this, and then forever get it. But I was not one of those people. It took me quite a while too. It even got down to, I think, I don't know, like week 10, right before the third exam, that I I messed up reading a cell arrangement. You know, it happens. It was a tetrad. I'll give myself that. It was a tetrad and I thought it was diplo, but I digress. You have to look at the greater population. Any questions on this stuff? I mean, this is probably going to be your best friend. If you want to draw it out, you can. That always helped me, was to draw it out. Probably in a little bit better arrangement than this. I would probably, you know, make it a pyramid scheme, like bacillus, diplobacillus, streptobacillus in one area. Then I would have coccus, diplococcus, streptococcus, tetrad, staphylococcus. That's what I would probably do. Because then I could make it to where it's like 1, 2, 3, 4, 5 for the coccus, and then bacillus, I can make it 1, 2, 3. So how are we feeling? I'm gonna have to look up some like test stuff, like pictures that I could test you guys on, because I know some of it's, you know, not exact. Yeah, no problem. And again, like, you guys come up with so much better stuff than me. I sometimes am very far-fetched. It's like, it's the biggest leap in like thought. And I'm like, this is how I remember it. And then you guys are like, what is happening? But with so many minds in one place, we can come up with so many things. So we explained this a little bit, I believe, Monday, Tuesday. We explained this a little bit Monday, Tuesday, but is there any confusion with preparing a specimen or making a smear? Did we want to go over this momentarily? Or is it pretty straightforward?
Can you go over it?
Yeah. So what we'll do in the classroom is we'll take a glass slide and we'll appropriately label it. It's usually just a circle. And in order to tell yours from another person, it's LK. Like for me, I put my initials. It doesn't need to be a whole lot, because the thing is, once we use these, we can't keep them. Once you make a gram stain and you put it in oil immersion, they're basically done and they have to go in the Sharps container after you view them. You can't go back to another objective. It's oil immersion, done. Like, you put the platform all the way down and you toss the specimen. So that's why it's really important to always take pictures. You can take one at, you know, right before you go into oil and when you're cold. But more than likely, it's whatever the highest power she wants you to go to. So is what you'll submit. You won't always be submitting pictures, but it'll be here and there. But in order to prepare a smear, we're going to on one side do the Sharpie and then flip it over. We're going to do the non-Sharpie side. So everything's going to look flipped to you. And that is what we want. We want it to look backwards, because part of the process for staining for the gram stain is going to be using ethanol. And ethanol is, it takes off Sharpie very well. So we don't want to start gram staining and, you know, pour ethanol on this and all of a sudden all of our Sharpie's gone. And not only that, but the dye could run into the bacteria. So we don't want discoloration, and you know, we don't want our Sharpie marks all of a sudden be gone, because then we can't see what our border was and where we should look. So we flip it over so that we have a blank side, right? So when we can see through the glass and we see that circle, we're going to put 1 drop of distilled water. It's going to be distilled water. So when we put this drop of distilled water on it, this slide is ready for bacterial contact. You're going to take with your incinerated loop even less than what you would do for a streak for isolation. It's a very minimal amount. Like she said here, the left and right, way too cloudy. If you try to dye that, it's going to look like a purple blob and you'll never be able to see individual bacteria. It's going to be bacteria on bacteria on bacteria. You're not going to be able to see through it. But on the middle, it's a little bit better. If you were to dye it, you would have to go towards the outside of that circle in order to even see anything. So less cloudy than the middle is what you're aiming for. It's okay, but less cloudy. So you're going to take, you know, maybe even like a quarter of a colony. It's not going to be a whole lot because remember, there's millions of bacteria in that one colony. So we want to take a little less because less is more in this situation. And then you're going to put the bacteria into the drop of water on your slide and just switch it around in the limits of the circle that you made. And it's not going to look like the water is spreading because it's, you know, its surface tension isn't going to allow it to spread evenly across the slide. But bacteria is going to stay there. It'll be a little more concentrated in the water, but it's going to spread in that circle. So once you spread your bacteria, you're going to put it on the heat plate and it's going to heat fix it to the slide. It's not going to move anymore. It's going to be dead and we'll be able to stain it that way. And of course, after you spread your bacteria, you're going to fling your loop again. I hope you guys all know that you're maintaining as sterile of an environment as possible. So this whole air dry and flame thing, don't worry about it. It's not going to happen. So like I said, it's going to, heat fixation is going to attach it to the slide. It's not going to rinse off the microbes because remember what happens if we wash our hands? The microbes are going down the sink. So with the glass slide, it's even slippery of a surface. If you swabbed or saw somebody swab a metal surface during our first streak for isolation, you saw that they might not have grown as much because the bacteria simply like to fall off no matter what. Even though a lot of people touch it, they do just fall off. And remember when you're heating these guys, you want to make sure that all the water has evaporated, but you also want to make sure that it hasn't sat there for more than probably 7 minutes. I think 7 minutes is like the max that you can go. So with that 7 minutes, you don't want to go past it because then it's starting to lyse the bacteria. Lyse isn't a good word. It's going to start degenerating the bacteria and it's not going to look the same as it would had you waited less time. Okay. Okay, let's see. So the whole point, I'm going to skip past this. If you guys are just, you know, like, okay, I'll just memorize this stuff. Is that okay? Or is there any theory behind Menevil's, Schaefer-Fulton, and the Zeal-Nelson acid fast? Is there any theory-based stuff on those you want me to go over? Or do you want me to just jump into Gram-stain? Because remember, this is for you guys. I could talk your ear off all night about some of this stuff. Because this stuff, you'll never do these three in class. You'll only ever be responsible for the Gram stain. That's the only stain we do. Unless something has changed, but that's the only stain I ever do. Okay, straight to Gram stain. Cool. So the Gram stain. What are we looking for with the Gram stain? Well, we're looking for can we go over what we need to know about them? Okay, yes. Briefly, I'll let you guys know. So what you'll need to know for each of these is the name, what they differentiate for, what's the first stain, and what's the counter stain. Okay. And then kind of maybe like one other special fact. Basically, you know, for this one, I'll need to know that it's Menevil for the capsule. It's differentiating if a bacteria has a capsule or not. I need to know that the primary stain is carbonyl fusion, and how it acts. And then I need to know that the counter stain is Congo red. Does that make sense? So this one, it's capsule. This one's for endospores. And then this one's is for waxy cells. It's going to be different cell wall. Okay. So that's basically all you need to know for each of them. is how they differentiate, what's the first stain, what's the counter stain, and usually there's like a special tidbit. So like these are for waxy stains, waxy cells in specific. What do? What are some waxy cells? Okay. All right, now we'll get to the gram stain. So for the gram stain, the first step is to apply the crystal violet. That is going to be our purple. Remember, violet is another word for purple. So that is going to be our primary stain. Now, when we go back, we're going to, apply the crystal violet, and then we're going to wait a few seconds. And I think the specifics, it's not 1 minute. I believe that it's 10 seconds for the primary stain, the crystal violet, and then you'll rinse, and then you'll apply the mordant. And the mordant, remember, if you ever, like, I don't know, I know on TikTok for me, sometimes I get those bricklaying videos and they're very satisfying. But what they're using between the bricks is the mordant. That's what keeps it in place. So the mordant is iodine in this situation, and the iodine is keeping the crystal violet in place. But it's only for gram positive that it's going to be able to keep it in place. Okay, so we're going to apply the mordant for 20 seconds, and then we're going to rinse it with the water, and then we're going to decolorize. Now, with the decolorizing stage, this is the ethanol that I was talking about that is going to wipe off your Sharpie. The decolorizing stage is going to take that crystal violet out of the gram negative. Okay, so it's going to make those clear again. But the gram positive are going to continue to stay purple. So it's going to be 10 seconds with ethanol, and then we're going to rinse off with water. And then we'll apply our counter stain, safranin. And that's going to give our gram negative cells that pink color, and then they'll stay with that pink color. So that's why it says imparts color and decolorized cells. Okay, does that make sense? Have I lost you guys? Because I know I'm kind of going through this a little fast. I don't mean to. I'm sorry. Okay, perfect. Okay. So when we apply our counter stain, it's going to make our gram negative pink and keep our gram positive purple. So that's how we differentiate cells. Okay, so those are going to be your basic steps. I know this slide says a minute and things like that, but I promise you it's not like that. And then afterwards, we're going to kind of do the same thing we did when we did our initial smear. We're going to put it back on the heat plate so that the water and any excess material can evaporate, and we have a clean slide. So I think that's the end of the slideshow. So we'll put it back on the table, on the heat plate, and then we'll be able to view it under the microscope. And how cool is that? Whatever you grew in your house, you're going to be able to see under a microscope. I think that's pretty cool. I know I was able to see my own strep. I was able to see because I swabbed my water bottle, and I was able to isolate 2 different colonies. And I think I still have the gram stain of both of them, but pictures wise. But it's going to be very interesting. I encourage you guys to, whatever you swab, make sure that you take a picture and then show it to everybody at home and say, this is disgusting. It's not disgusting. We all have bacteria. It's everywhere. So we live in harmony. But that's what our gram stain is going to be. And I'm going to say that gram stain is probably one of the, I really enjoyed doing all of this. It's super cool. But gram stain is going to be one of those fundamentals. It's always going to be there because, like, the Zeal Nelson, that's going to be for your waxy cell walls. And there's not a whole lot of bacteria with waxy cell walls. It's a very niche thing. Same thing with endospores. Not all bacteria have endospores. What's the other one? The last one is capsules. Not all bacteria have capsules. It's either capsule or not, but the thing is most bacteria are either gram positive or gram negative, and that can tell us a lot about what they do and how they function and how what's in their cell walls and how pathogenic they are. And then we also can use this information to identify unknown, wink wink, unknown bacteria. I'm sure that you guys are going to be getting some kind of unknown bacteria, wink wink, and gram staining it. So is there any other questions that we have on the gram stain? I know it's a preface. You guys will learn a little bit more about how it works on the molecular level, kind of like on a more granular level, about how the mordant works, how the decolorizer works. There was a really good video that we were shown last year, and if she doesn't show you a video in class, let me know and I'll look for it. Was this a good preface for you guys? Okay, I got 1 thumbs up. Cool, cool, cool. 2, yay. Okay, okay, cool, cool, cool. I'm glad. Wow, everybody's interacting. Yay. Okay, so I'm gonna go ahead and stop the meeting for just a minute. We'll reconjoin. I want to give you guys a full 5 minutes. So we'll reconjoin at, if I can do math, 7:42. 7:42, because I'm gonna probably talk for another second. 7:42, and we will jump back into whatever you guys would like. If you guys want me to review some microscopes, I know I did that on Monday, but if you guys are still having any trouble with it, let me know. If you guys go get a drink of water, stretch, do whatever. But if you have any other questions, we'll reconjoin in 5 minutes. So I'm going to end the meeting, and I will see you guys momentarily, okay?